Welcome to the 2021, having a hard time with that, Geology Lecture Series at SWOCC, gone virtual. What interesting times. About 6 months ago, today's speaker was scheduled to be in Coos Bay. We all know what has happened since, and I'm pleased that we've been able to reschedule Dr. Edgcomb in this new format, which, like most, both Ginny and I much prefer the in-person events and look forward to the time we can return to those good old days. That said, for over 20 years, I've been hosting scientists here at the edge of the known world, center of the universe, and they've delivered cutting-edge scientific research to our students and community. I believe this is important now as ever, and I'm pleased that we have some fantastic speakers lined up for live stream talks this year. This includes a really interesting twist for the 16th annual Cascadia Anniversary Lecture, Whale Seismology, with Dr. William Wilcock on Tuesday, 1/26 at 7 p.m. I don't get as many questions about the new building. I guess that's because I don't see many people right now, but it's down to finishing final details in the punch list. So we'll be starting the move-in fairly soon. This is the last talk before 2021, and many groups ask for your financial support at this time of year. I know that many of you have contributed to the lecture series tip jar over recent years. You can still donate to the lecture series by sending a check or donating online through the SWOCC Foundation, www.socc.edu, or consider funding college scholarships awarded by our foundation. They're going to be as crucial as ever over the coming years. I'm pleased to introduce today Dr. Virginia Ginny Edgcomb from Woods Hole Oceanographic Institute, an ocean discovery lecturer. She earned a BS from the University of Virginia and a Ph.D. from University of Delaware, completed 2 postdocs at Woods Hole. She is also author or co-author in over 75 publications in leading scientific journals, and is associate editor for the journal Extreme Biology and also a review editor for Aquatic Microbiology. Ginny's talk will address one of the last frontiers for biological exploration on planet Earth, the lower oceanic crust. We're also trying something new. We just recorded the talk, so Ginny will be able to answer questions live in the chat box during the talk, as well as following the presentation. Ladies and gentlemen, it's my pleasure to introduce Dr. Ginny Edgcomb with Life at the Edge of What is Possible, Microbial Biosignatures in the Lower Oceanic Crust.
So thank you for inviting me to share with you some of our ongoing and recent work exploring life in the lower ocean crust. It's a real honor to talk with you today, and it's a shame with the pandemic that I can't be there in person, but hopefully I look forward to talking with some of you during the Q&A session. Next slide, please. So I'd like to start today with an introduction to the International Ocean Discovery Program, the IODP. This is something that they've asked me to do in each talk. And then I'll move quickly into a discussion of Expedition 360, which explored the lower ocean crust at a site in the Indian Ocean called Atlantis Bank. I'll talk about our detection of the distribution of cells and of biomarkers of cells and the stories that they tell us. And I'll show you some of the activities that we measured, and I'll end with a brief story about deep biosphere fungi. Next slide. So scientific ocean drilling has a long history. It started with Project Mohole, which segued to the deep sea drilling program, and then the ocean drilling program, and then the integrated ocean drilling program, and finally in its current incarnation, the International Ocean Discovery Program. Next slide. These programs, as you can see from the colored dots that correspond to the different programs, have sampled a wide range of sites in almost every major ocean basin. And from these samples, we've learned a lot about deep biosphere processes and of course about earth processes below the seafloor. Next slide. The IODP program uses multiple platforms, including the JOIDES resolution that you see there at the top, which is the ship that I sailed on for Expedition 360. It's operated by the US. The CHIKU, which is operated by Japan. These two ships differ because the JOIDES is not a riser-based platform, which means it's not directly attached to the seafloor except by the drill pipe. And then there are many mission-specific platforms that are operated by an international consortium, and they're chosen based on scientific objectives. Next slide. We need well-trained geoscientists now more than ever before, and this is because humanity is facing a lot of issues that you're well aware of that are addressed by geoscientists. These include mineral and water resource needs, energy needs, environmental degradation, natural disasters and hazards, climate change, sea level rise, and coastal erosion, just to name a few of them. Next slide. So you can participate in the IODP program by applying to sail as a shipboard scientist or technician. You can serve on IODP science advisory panels, the US Science Advisory Committee, and you can request samples and data for your own research. Graduate students have many opportunities. They can apply to sail. There are fellowships available for graduate students, and you can assist in post-cruise research. And you can learn more about all of these things by going to usoceandiscovery.org. Next slide. So today's talk is going to focus on microbial life. And I just want to point out that microorganisms are the oldest forms of life on Earth. Our oldest fossil record of life on Earth comes from stromatolites, which are built by microbes. And there are stromatolites that are dated back to 3.5 or so billion years ago. Next slide. Microorganisms are found in all three domains of life, as you're well aware of the bacteria and archaea, and people sometimes forget that the eukaryotes, where humans are found within animals, as well as fungi and plants, many of the lineages of eukaryotes are microbial, including almost all of the deepest branching lineages. Next slide. So some trivia for you about marine microbiology. Approximately 50% of the oxygen we breathe comes from marine phytoplankton. Next. Methane and other greenhouse gases are produced by microbes in subseafloor sediments. Next. And not just by organisms on land. Next slide. They're involved in the bioremediation of chemical spills. Next. And they shape ecosystem processes. Next. And they exhibit all trophic modes. And they are heterotrophs, they're autotrophs, there are decomposers, and there are parasites. Next. So some major questions asked by marine microbiologists include: what is the biomass and activity levels of microbial populations that are present in different habitats? Next. What is their diversity and their function? Next. How does habitat chemistry influence their population structure and their activity? Next. And how do their activities influence aquatic subsurface biogeochemistry? So exploring the extent of the deep biosphere has implications for our understanding of carbon and other nutrient cycling on Earth. And because of the sheer extent of its volume, even if life persists in the subsurface at very low abundances and low activity levels, this can translate to a significant impact on pools of carbon, just as an example. Next slide. So what is the deep biosphere? This is somewhat operationally defined, depending on who you ask. You get a lot of different definitions, but We'll start with the idea that it starts somewhere between 1 meter and 10 meters below the seafloor, and it depends on who you ask. It's basically anything that's too deep to be sampled without specialized equipment, such as gravity cores or a drilling ship. Next slide. So the deep biosphere, in this schematic, sort of gives you an overview of the different layers. In most places in the ocean, the seafloor has abundant sediments, and these sediments can be up to thousands of meters thick. These are typically underlaid by lavas, basaltic rocks of the upper crust, and sheeted dikes. And these are underlaid by the gabbro rocks and peridotite tectonites of the lower crust. And some examples of these rocks are on this slide in places on Earth where they've been exposed above land. At the top left, you see the basaltic pillow lavas. Top right, the sheeted dike complexes. Bottom left, the peridotites, and bottom right, the gabbros. And during Expedition 360, we sampled almost exclusively gabbros. Next slide. So IODP Expedition 360 sampled a site in the Indian Ocean called Atlantis Bank. And the reason why we sampled here, which was southwest of Madagascar, as you can see in the lower, on the left-hand side, the reason for sampling at this site is because the lower crust has been rotated up and exposed at the seafloor in only about 700 meters of water. And so this allows a drilling ship to drill directly into these gabbros without having to drill through extensive sediments or overlying basalts. Next slide. The microbiology team consisted of myself, Gaetan Bourgou at the University of Brest, a marine mycologist, Jason Sylvan from Texas A&M, who sailed with me on this expedition, Florence Schubatz at the University of Bremen, Frieder Klein at Hui, Jingtao Li from Tongji University, Vivian Mara from Hui, and of course the stars of the shows, the microbes themselves. Next slide. So this is a quick video for you that will show you the operations on the drilling floor of the ship. You can start the video, please. So the ship never sleeps. It operates 24/7. This necessitates the science party to divide itself into two parts so that we can work around the clock. In this short video, the drillers are doing what's called tripping the pipe, which means bringing it up in sections so that they can change the drill bit. And the cores, the rock cores, are brought up in 10-meter sections when core is retrieved. And in the next slide, shows you the drilling bit that we used, which is the rotary core barrel at the top left and top center. And you can see that the center of the drill bit retains a core of material that the bit is drilling through. So this column of rock, which you can see on the bottom right image, is brought up to what's called the core receiving platform, where it's logged and labeled and then divided into 2-meter sections. Next. So in the deep biosphere, or below the seafloor, life has many challenges, aside from pressure and of course, increasing temperature in most sites with depth. Life requires sources of carbon and reducing equivalents, or electrons, as well as electron acceptors. And possible sources of these include seawater that is entrained into the crust from above, as well as hydrothermal fluids from below that circulate through the crust. Okay, the entire ocean volume is thought to circulate through the oceanic crust about every 10 million years. And this underscores the tight connection between the oceans and the oceanic crust. Water moves through subseafloor basalts and gabbro aquifers, through cracks in the rocks and fissures, And in the rock cores that we received, you could see carbonate and clay-altered veins in some of the rock samples, which is evidence of previous flow or current flow of water through these rocks. Additionally, rock-water interactions such as serpentinization or the pyrolysis, which means the heating of organic matter, can release compounds that can be used by life for carbon and energy. Next slide. If we use serpentinization as an example, this is the hydration of olivine. Olivine is the rock on the left. And olivine and seawater and carbon dioxide react together to produce the rock serpentinite on the lower right. And in this process, hydrogen, methane, and short-chain alkanes are released. And these are fantastic energy and carbon sources for many types of microbes. Next slide. You can start this video. So this shows you the core receiving platform. So we get an announcement about every 45 minutes that a new 10-meter section of core is brought up on deck. It's labeled, sectioned, and divided into 2-meter sections, which are brought inside. The geologists image these sections in a myriad of ways that I don't have time to go into. But immediately after, next slide, after bringing these sections inside, a piece is selected for microbiology very quickly. And this is a photo of me and one of the co-chiefs selecting a sample from one of the 10-meter sections, and I prioritized samples that showed evidence of alteration of the rock, where fluids had changed the mineralogy of the rock. I was looking for rocks that showed evidence of veins or cracks, voids where microbes would be likely to inhabit if they had access to fluid flows. Next slide. So Jason and I ran a tracer in the drilling fluids that are used during this operation so that we could monitor for our adequate removal of any exterior contamination from each sample that we worked with. After cleaning the exteriors and sterilizing the exteriors of the rock samples were brought inside a walk-in laminar flow work area where they were smashed up into subsamples for many different types of analyses in a sterilized rock box. Next slide. And this is a down core image. On the left-hand column, the colored column, you can see the colors just represent different types of gabbros that we drilled through. From top to bottom, you're looking at samples from the seafloor all the way down to almost 800 meters below seafloor. And the red dots in the second column, the tiny red dots, show you the depths at which we collected samples for microbiology. The next column, the third one in, with the dark green bars, is showing you samples that Jason and I were able to successfully measure the molecule ATP. And ATP is a high-energy organic molecule. It's a universal mediator of cellular activities, and it provides energy to drive cell processes, and it's a precursor to DNA and RNA. It's one line of evidence for samples that might contain living cells. We also measured another enzyme, alkaline phosphatase, and alkaline phosphatase is a widespread enzyme. It's involved in dephosphorylating compounds. I'm not showing those data here in this slide. Next. So in addition to that, we performed nutrient analyses, carbon, hydrogen, nitrogen, and isotopic analyses. We analyzed lipids from each rock sample, and we performed cell counts and enrichment culturing of prokaryotes. The cell counts were conducted in Jason Sylvan's lab in a clean room facility, and the fungal culturing was conducted in my lab and in Gaëtan Bourgou's lab in France. Next. We also extracted DNA and RNA from all samples for retrieving genomic DNA and data on expressed genes. That was done in my lab. And we performed scanning electron microscopy and Raman microspectroscopy on thin section samples that we produced for each rock sample. The bars on the right two bars, the green and the blue vertical columns, show you data from the geologists during the expedition on the intensity of carbonate and clay-altered felsic veins, respectively. Those, again, are signs of alteration in the rock and where either water was flowing currently or previously through those rocks. You'll notice that the top of the site that we drilled had very little alteration, and you'll see in later slides that that corresponds to the lowest recovery of signs of life, and that there were more signs of life in regions of the drill hole where there were higher signs of alteration and faulting. Next. For messenger RNA analysis, this was a very laborious process to extract RNA from these rock samples. We had to prioritize about 11 samples. That's what we figured we could manage. And our criteria for selecting samples for this was based on the samples that had the highest degree of faulting intensity, the highest degree of alteration and presence of carbonate or felsic veins, and information from the carbon, hydrogen, nitrogen, and sulfur analyses of these rock samples. Next. So how do we detect cells in these gabbro samples? Next. This work in Jason Sylvan's lab starts just briefly with crushing the rock in a clean room facility, extracting it with a mixture of formalin, detergents, and methanol, shaking and sonicating, and then loading this material onto a density gradient, which is a material made out of a material called nicodens. These tubes are spun, and the cells are retained in the light fraction in light gray. And the column material is re-extracted a second time, and all the gray fractions are pooled together and stained with a stained cyber green, which stains DNA of cells, and filtered onto a filter and observed with a microscope. Next. So the cell counts that we obtained were very, very low, and also variable. And for context, cell counts in the global sedimentary subsurface are thought to be on par with the counts in the entire ocean volume. But the biomass that we detected in the lower crust is at the very lowest end of those nutrient-poor sediment samples, and it's very, very, very heterogeneous. Next slide. So I want you to see if you can see the cell in this image. Okay, there is one cell in this image, and this just illustrates how difficult and time-consuming this cell counting is that was done by Shuying Wei in Jason Sylvan's lab. If you hit the next slide, there's a zoom in now to an area that you should be able to see the one cell there, yep, that's lit up, that is truly labeled with the cyber green. Next slide. Takes a lot of patience. So at the end of the day, after counting many, many replicates for each sample, the average cell density turned out to be around 6.5 times 10 to the second cells per cubic centimeter of rock. And this was very close to our limit of detection. It ranged in some samples from undetectable, as you can see, very much so near the top of the drill hole, to 1.73 times 10 to the fourth in other samples. There was a very strong positive correlation between our cell counts and the frequency of felsic and carbonate veins. Next. So what can DNA and lipid biosignatures extracted from these very same samples tell us about these cells that we saw under the microscope? So we'll start with the lipid biomarkers. Next. So after cell death, the labile heads of membrane lipids are the head group on the left there in yellow is enzymatically cleaved. And this leaves the core lipids on the right in blue, which can be preserved over millions of years. So that's why geologists use lipid biomarkers to examine the geological record. The ratio of intact polar lipids that we detected to core lipids, and this again is work done in Florence Schubat's lab at the University of Bremen, supports their in situ source and the minimal accumulation of fossil material in these samples. Next. So the way this is done, again, in a clean facility, the rock is powderized and lipids are extracted using a modified Bland-Dyer extraction. And due to the very low expected biomass, Florence used a targeted and ultrasensitive approach that combined ultra-high pressure liquid chromatography and coupled that to triple quad mass spectrometry, as well as multiple reaction monitoring experiments that are designed to look for specific compounds of interest rather than profile all the lipids that she could see. Next slide. So the lipid controls, were run for each of the samples she analyzed. Any peak that was detected in any of the controls was subtracted from the sample. There's just one sample to give you a representative of this on the left, and then its corresponding blank on the right. And if you look at the y-axis for the two, you'll see that the amount of lipids detected in the blanks was very much lower than in the samples. Next slide. So bacterial glycosidic dietherglycerol lipids, that's a mouthful. We're just going to call them DEG lipids for now. Were detected at all depths. And DEG lipids are not normally found in oxic water column, which is reassuring. So we weren't looking at seawater lipids. They're not normally found in the water column or in oxygen minimum zones. But they are detected in marine sediments, cold hydrocarbon seeps. They have been seen in the Black Sea and Cariaco Basin sediments, and they require less cellular maintenance than fatty acid containing lipids. And we can't prove this, but it could be that the use of DEG lipids may be an adaptation to the stresses of living in low energy plutonic rocks. Again, these are core lipids, not intact polar lipids. Next slide. Next. Okay, something kind of went fluey with the last slide, but we'll just keep moving on here. The DEG lipids that were detected are typical of mesophilic bacteria, and the potential sources of those include the Aquaficiae, firmicutes, and some sulfate-reducing bacteria. And these were detected in our messenger RNA pools that I'll talk about later. And Florence is in the process of designing screening techniques to look for the intact polar lipid counterparts of these bacterial DEG lipids that should be more representative of the viable community. Next slide. So looking at the archaeal lipids, she does have data here for intact polar lipids of archaea. They were dominated by archaeol-based lipids over glycerol dialkyl glycerol tetraethers. This is distinct from what is observed typically in deep biosphere sedimentary settings. And this suggests that perhaps in the lower crust, there's an indigenous archaeal community. Next slide. The dominance of archaeol-based lipids over GDGTs was observed another time at Lost City. And Lost City is on the Atlantis Massif in the Mid-Atlantic Ridge, and also in some anaerobic methane oxidizer dominated cold seeps. Next. So the DNA-based assessments of diversity give us a picture of a low diversity community that includes not just chemoautotroph taxa, but heterotrophic taxa, many of which are described from deep sea and polyextreme habitats. And there was increased diversity in samples that had higher faulting and alteration zones. And for the DNA-based work, we ran a lot of controls and subtracted any sequences that were in control samples from the DNA extraction kits, as well as the drill fluid controls. Next. So the variation in taxonomic composition among the samples is actually reassuring to me, and it suggests the availability of carbon and energy substrates is also heterogeneous at different depths. If we were recovering the same diversity in every sample, I would be a bit more worried. Next. The SAR-324 Delta proteobacteria were detected in many of the samples. These are known to have metabolic flexibility, including the ability to oxidize alkanes, methane, and or sulfur for energy. Next. And the detected DEG lipids that I mentioned a few minutes ago could be from some of these Delta proteobacteria. SAR-406 and Marina microbia were also detected in all of these samples, and these are putative sulfur and hydrogen oxidizers. But of course, it's important to remember that DNA extractions are not proof of active microbes. Next. If we look at the distribution of these marker genes, these DNA-based marker genes in light of the environmental variables that we were able to measure, we see that the diversity is best explained by or correlated with concentrations in ATP that we were able to measure, as well as fracturing intensity and graphic lithology, which means the type of rock, how highly altered it is. So fracturing and alteration locations are where seawater from above or low temperature fluids from below can most readily permeate the rock. Next. Now, Bobarker-Jorgensen published a beautiful paper in which he reports that most subsea floor microbial cells are likely to be on physiological standby. And this is based on culture-based studies of isolates obtained from lots of different types of samples, nutrient-rich samples in blue and nutrient-starved samples in red. And if you look carefully at this graph, you can see that the red dots, okay, the metabolic rates that he calculates for those red dot samples, the nutrient-starved samples, equate to doubling times or turnover times, how long it takes a population to double in its size, okay, of 1 year to hundreds of years. And this means they're like barely eking out a living, barely staying alive. And so we were very curious to see whether we would be even able to detect any signs of life in the lower crust. And our ATP and our enzyme activity and our messenger RNA activity suggest that not all cells in the lower crust are on physiological standby. And of course, in sedimentary environments, this is the case as well. Next slide. So we provide the first insights into in situ activities within the lower crust by examining the messenger RNA pools that show us that some cells are active. And this is supported by enzyme activity and ATP measurements. Next slide. The way this works is, again, under very clean conditions. The rock samples are crushed, they're extracted. The RNA is amplified using a kit because it's in very, very low abundance. And it is sequenced and assembled and subjected to many different contamination controls that require us to subtract any data that we recover in drill fluid controls. Next slide. Okay, so in drill fluid controls, laboratory contamination controls, controls for contaminants that can be brought in by molecular kits, and all of those data were subtracted if they appeared in any of our samples. Next slide. So what did we look for? To find basic proof of living cells, we focused on core functions. So all cells require sources of carbon and energy, such as carbohydrates. They require amino acids for protein synthesis, fatty acids for making membranes, and nucleotides for replicating their DNA and RNA. Next. This is a very bird's eye overview of the downhole recovery of expressed genes in 6 different categories of metabolism. And we'll start with the gray portions of the bars, which are amino acid metabolism, which was detected in almost every sample, evidence of amino acid synthesis or degradation. And the degradation of amino acids would imply their use as a carbon source. Okay, next slide. So branched chain and aromatic amino acids can fuel central metabolism, okay? So if they're used as a carbon source, they can fuel central metabolism by enabling production of a molecule called acetyl-CoA, and thus the Krebs cycle if oxygen is present. And you may be thinking, well, why would oxygen be present? But remember, seawater can be entrained through these fault zones, so it is plausible that at least sometimes there can be exposure to some oxygen. In the absence of oxygen, their breakdown yields short-chain fatty acids and eventually CoA thioesters that can serve as electron acceptors under anaerobic conditions. We detected the expression of genes encoding enzymes involved in the metabolism of 14 different amino acids with different properties, including the 3 aromatics, tryptophan, tyrosine, and phenylalanine. Next slide. So these aromatic amino acids can come from a pathway called the shikimate pathway in the red box to the left, which we detected expressed genes for. Next. And they can also come from the degradation of polyaromatic hydrocarbons. And we detected expressed genes for the degradation of polyaromatic hydrocarbons. And polyaromatic hydrocarbon degradation can facilitate the production of aromatic amino acids, if you think about it, because energy is conserved by avoiding construction of that aromatic ring, which is a very expensive ring to build. Next slide. We did detect the genes for degradation of complex hydrocarbons, these aromatics, in 8 of our samples with the arrows. These are the tan bars, and the arrows point to the samples where these were detected. Next slide. And we have expression of genes for the uncharged amino acids serine, threonine, and glycine, and branched chain amino acids, leucine, isoleucine, and valine. Next. And some anaerobic bacteria are known to use Stickland reactions. These are reactions that ferment pairs of amino acids to release energy. And in these reactions, one amino acid acts as the electron donor and one as the electron acceptor. These processes have only been demonstrated in laboratory experiments, but our mRNA data suggests the production of known pairs for this process, so it's possible that this may act as a source of energy in the lower crust. Next. We have genes expressed for glutamine and glutamate synthesis, and these are two important amino acids involved in recycling intracellular ammonia and coping with oxidative and osmotic stress. And glutamate is also the basic substrate of about 80% of non-essential amino acids. Next. So the lipid biomarker data combined with the mRNA data provide evidence for the synthesis of fatty acids, and transcripts for the beta oxidation of those suggests their use also for carbon and energy. Next. The mRNA also indicates phospholipid membrane synthesis as well as peptidoglycan synthesis, and these are two lines of evidence for at least the presence of some cells that are reproducing or repairing. Next. The expression of genes involved in guanine, adenine, and uracil synthesis, as well as thymidine, shows you that production of nucleotides is also an active process. Next. So physiological processes combine genes for Krebs cycle as well as chemoautotrophic carbon dioxide fixation. And these are in dark blue, and we detected 5 CO2 fixation pathways in our samples. Next. So carbon storage, this is something I find interesting. And as an adaptation for the deep biosphere, carbon storage appears to be an important strategy for supporting life here. And this is because we have quite a lot of expression of genes associated with the degradation of complex polyaromatics and amino acids that can provide acetyl-CoA and propionyl-CoA that you see in the red boxes, okay, as their final products. And hit next, please. Okay, these two molecules can fuel basic metabolism, but they can also lead to the production of carbon storage compounds called polyhydroxyalkanoates. And these are in the blue boxes that you see toward the center, left and right. These polyhydroxyalkanoates are storage molecules. The beta oxidation of fatty acids at the top in the green box can also produce these polyhydroxyalkanoates. Next. Polyhydroxyalkanoates are produced by bacteria that are grown under nutrient-limiting conditions when excess carbon is provided. This has been shown only in laboratory experiments, and it may be an adaptation to living in the nutrient-poor deep crust, where if you see carbon, you better grab it, because you don't know how long it's going to be till you see it again. These molecules are synthesized via the Fa operon that encodes 3 genes whose expression we saw in our 2 deepest samples. Next. So microorganisms in these rocks can also be cultivated. In the lab of Jason Sylvan, they conducted some intriguing experiments, work done by Shuying Wei, next slide, where she added different combinations of nutrients. Next slide. So she had a no treatment control for every sample she ran this for, where it's just rock material and artificial seawater. She added nitrogen to the same sample in the form of ammonium chloride. She added nitrogen and phosphorus by also adding potassium phosphate, and she added carbon in the form of a combination of lactate, acetate, and formate. Okay, and then at periodic intervals, the headspace was analyzed to examine whether they could detect methane or other gases. And I'll talk just about the methane next. So at 2 different intervals, after 25 and 60 weeks, they detected methane production. The blue bars are after 25 weeks, the red bars after 60 weeks. The treatment did not have a significant effect on the methane production, next, or the taxonomic composition of the community that they recovered. There was a weak correlation with vein frequency and methane production, next. But unfortunately, they were not able to recover the archaeal 16S marker genes that would support methanogens. If you look at the literature, though, recovery of methanogen 16S genes is quite often problematic, so this is inconclusive. Next. And so what they're doing is they're doing an isotopic analysis of this headspace methane to see whether it's biogenic or abiotic, because other possibilities for the methane could be that they are coming from inclusions, fluid inclusions in veins that have methane in them. Next. And next, you can advance again. So now to move finally to the last part of this story, which is the fungal culturing efforts. And these people don't often think about fungi in the deep biosphere, but Gaetan Bourgou's lab has done a lot of work in this area. His graduate student, Maxence Kamenar, used our messenger RNA data to look at the fungal transcript data and to see whether there was evidence for dominant pathways and affinities for certain substrates that might help them optimize their culture media. Next. They use a very high throughput approach. And so when looking at our mRNA data, it was a little bit difficult because the coverage, these, if you look down from top to bottom, these are just different categories of genes that were expressed. And in these dot plots, you'll see that not all samples, the samples are, the shallowest sample is to the left and the deepest sample to the right. Not all samples produce detected genes in every category, and this could be due to incomplete or inadequate sequencing depth for these samples in order to cover the fungal community. It could be an artifact of our extensive data subtraction, or perhaps excessive data subtraction to avoid discussing contaminant data, or it could reflect the actual biological distribution. The red box is around one sample from around 228 meters below the seafloor that had quite good recovery from almost every category. And this was again in a sample in a high degree of faulting. Next. So they used a method that is similar to a spectrophotometer. It's called MALDI-TOF-TOF mass spectrometry. And they use a 96 or 384 well plate format with different media in the different wells, and the samples applied across these plates. And the wells that produced growth are detected by the determination of scattered light over time. And wells that produce growth, they had to determine whether the growth in those wells was unique or not. And to do this, they used a method called MSPCR, which is bisulfite sequencing. And this method, you can read more about it, determines patterns of DNA methylation. And they also used the method of MALDI-TOF-TOF mass spectrometry, which is a rapid, accurate, and sensitive technique for providing molecular weight information on what's in that well in terms of proteins, peptides, oligonucleotides, nucleic acids, and carbohydrates, for example. Next. Unique cultures were then, of course, identified with marker gene sequencing. Now, the idea of fungi in crustal samples is not a new one. There was evidence of fossilized fungi in carbonate veins and vesicles from Eocene basalts. This was published by Iverson et al. In 2012. And in these pictures at the top right, you can see typical fungal filament morphology, and down the golden picture there at the sort of towards the bottom left shows a characteristic branching anastomosis, it's called, and you can see where two filaments are connected to one another by a branch that connects them in between. In the center bottom image there, the green one, you can see characteristic septa or spaces between the cells and filaments. Next. So Maxence is reporting here the downcore fungal diversity from shallow on the left to deep on the right. And without looking at the details of the different genera, which is what is depicted in the different colors, you can see that diversity is higher in some samples than in others. And the pattern is correlated with the regions of fracturing and alteration. Next. These are some pictures of some of the cultures on traditional plated media. I think they're quite beautiful, and it just illustrates the diversity of filamentous fungi and yeasts within this collection. Next. Maxence tested their metabolic capabilities of 26 of the isolates that he grew, and these 26 were isolates that showed clear preference for growing in the presence of marine conditions, 1.5 to 3% sea salts. He tested the metabolic capabilities using the biolog system. This is a microplate-based system that tests 96 different organic substrates. Unfortunately, the biolog system, you can't use salt, okay? So you'll see something happens here. The yellow is our cultures. So the cultures are down the y-axis. The substrates are across the x-axis. You'll see the yellow is no degradation. The red is high degradation. There's stars next to a couple of the isolates. A single star indicates the ability to use archaean lipids. 2 stars, the archaean peptidoglycan, and 3 stars, bacterial peptidoglycan. And I just point this out because if you're a fungus, a saprophyte, generally speaking, you're going to need a source. You know, what is going to be your source of carbon? And I'd just like to throw it out there that we think it's primarily necromass, so they're feeding off of the prokaryotic community and each other. All right, so it's logical to think that at least some of them would be able to access these very tough molecules, these peptidoglycans and some of the lipids. In the middle, you'll see a band of isolates that showed very little degradation. And these were isolates that absolutely would not grow without salt. Okay, so that sort of explains their inactivity here. Next. So looking for direct evidence of cells in the rocks is very difficult. We tried to use Raman spectra of organic inclusions, and we started with scanning electron microscopy. And when you're looking through the scanning electron microscopy, if you have it linked with the capability to profile or to look for carbon as well, you can see regions of carbon-rich inclusions. And we did image many of those, but we were looking for a needle in a haystack, a place where an organic inclusion was overlaid by mineral, by the mineral itself, because this would be stronger evidence of in situ origin and not something that was deposited on top of the rock or the thin section during thin section prep. And we did find one place where we have these organic compounds in this image here from one sample that are associated with ferrous iron oxide bonds, and the inclusions reveal a filamentous texture that's intergrown with the carbonate mineral, which is calcite, the host rock. And this evidence is stronger evidence for its in situ origin. Unfortunately, we don't know its identity. Next. So our major findings from these sample analyses are evidence for the presence of intact cells in the lower crust, some fraction of which appear to be active. They're heterogeneously distributed. They include all three cells from all three domains of life, and primarily in veins and fractured regions. This is on the basis of mRNA and detected enzyme activity. Next. The mRNA shows that the metabolisms that are active favor pathways that can store carbon and recycle it into central metabolism. This would make sense that anything living in such an austere environment would have to be an efficient recycler. Biosignatures of taxa include relatives of thermophiles that may be delivered from deeper, hotter regimes through the fractures, as well as aerobic and anaerobic heterotrophs and autotrophs, methanogens and halophiles. Next. And the rock inclusions showed evidence of, when Frieder Klein looked at the rock inclusions, he found inclusions of carbon dioxide, hydrogen, and methane, and these could fuel some fraction of the detected taxa, combined with periodic influxes of seawater or subsurface fluids through these fractures and veins. The lipid biomarkers support the taxonomic affiliations of the DNA and mRNA biosignatures that we detected, and we have culturable bacteria and fungi. Next. So I just would like to thank you for your attention. I hope that that you have found this to be interesting. The IODP has so much to offer in terms of future expeditions that you might participate in. There's so much still that we have to learn. I'd like to thank the two co-chiefs of Expedition 360, as well as the staff scientists and other scientists I sailed with, and my collaborators. So thanks very much, and I look forward to talking with you after you listen to this talk.
Thank you, Ginny. So I've got one question. I kind of mentioned that when we were originally filming. It's been probably more years than I want to remember, but I hosted Dr. Mary Higby Schweitzer, who found soft tissue in T-Rex. And I know that in that project, she had some similar issues to the things that you discussed about trying to prove or dispel that contamination is the source of the biological component. You hit on a few of the points pretty well, I thought. What's the next step in that? Because I know my students in introductory geology are going to automatically think, well, gabbro, that can't have fossils in it or signatures of biologic life, because you've told us that igneous rocks are high temperature, et cetera, et cetera.
Mm-hmm. That's a great question, Ron, and thanks for asking that one. Let's just say that when I was sailing on the ship, this was something that the geologists asked about universally, okay, to Jason and me. Because we're both looking at this from slightly different perspective. These rocks, as you said, were formed under extremely high temperatures, and the clay-altered felsic veins themselves, the original veins, were also formed at really high temperatures. So those temperatures would have been too high for microbial life. It would have sterilized anything that was present at the time of formation. So the microbes that are present now have been deposited into the rocks through fluid flows at a later date. We can't really say when or how long that they've been there, but they are just utilizing the cracks and fissures, the faulting zones. They're being delivered probably into this biosphere by fluid flows, and it's the fluid flows that help maintain them down there. And teasing apart what's a microbe that's able to just live or exist down there now versus something that may have been contaminated our samples through the very dirty drilling process itself was our biggest challenge. And all I can say is that we knew that the only way we were going to build a stronger argument, and at first I should say we didn't really know what we would find, whether we would find any sign of life there or not. And we were okay with the scenario of not finding life. But it's just, we decided that what we would do is look at the same samples using as many different methods as we could with the limited material we were allowed, because there's nothing that makes a geologist more unhappy than to walk off with part of their 10-meter section of rock, So every biological sample was a prize. But these samples were broken up into many different types of analyses, and by running careful controls for each of those analyses, and then looking at the results of all of these analyses, we feel confident that we can say that at least in some of our samples, there's signs of active living microbes. And we threw out a lot of data in an effort to try to be cautious. And I think that we'll only be able to prove ourselves right or wrong by doing other such studies in the future at other locations to learn whether this site is representative of the lower crust or not, or whether it's an unusual example because of the faulting and fluid flows that are there. Did that answer your question, Ron?
Yeah, and it's one of the things that I always like as a past Devonian conodont researcher. We know they're extinct, and there's absolutely no question they show up or they don't show up. But one of the things
Right.
to dovetail when you said that, hey, we're okay not finding anything, we always used to joke, and I know pretty much every scientific group, there should be the journal of non-existent results so that people don't always go back.
Yeah.
But somebody goes back and then they find the one thing that you missed. The one other, and I think it's a really important question that you pointed at near the beginning in the introduction with IODP and with science in general, is that we're at a point in time where there's going to be a large number of opportunities in science and the related STEM fields. And just out of curiosity, what led you down that pathway initially to get into sciences and to become a scientist?
I guess I was an observer of nature and an appreciator of ecology from a teenager. My parents were very influential, but they also steered me away from science initially, and I studied finance. And I got a degree in finance, and then I took a job for DuPont, and I instantly hated it. Okay, I was pretty good at it, but I only lasted a few years before I started going back to school to fulfill my science requirements for grad school, and I knew that's where my heart lay. I didn't know exactly what I wanted to do, but I wanted to make a contribution towards better understanding our oceans. And I ended up in Woods Hole. My choice of study was very much influenced by some professors that I had, who inspired me. I didn't really go into it knowing that I would be a microbiologist, but I've really enjoyed my journey, and the oceans are changing very fast. And so I get motivated by, the common denominator in my lab is oxygen depletion. So we study in my lab oxygen depleted regions of the world oceans and sediments. And that's kind of what gets me up in the morning, is trying to better understand, how oxygen depletion is shaping what goes on in these important ecosystems.
Thank you. It's something that I've been thinking a little bit more lately, that our science building was built in the 60s and pretty much outfitted in the 60s, and making that transition to the new building here in the coming months is something that I think we'll be able to go from having served our students very well in fairly primitive conditions to newer conditions for a 2-year school, and providing that foundation for them to be exposed to science and to move on. And I think that's one of the things that I'm understand, and I'm always amazed at the level with IODP and the interdisciplinary connections, which are crucial because we all work in our own little silos. But being able to have the top people in the game in biology, in chemistry, in different aspects of geology makes that, because no individual can find that. And I appreciated the connection to enjoying what you do. And that's one of the things that I always tell students is you have to find that passion, and that passion for what's going to strike you, what's going to make you happy, what's going to make you enjoy going to work every day. So for that, I've been fortunate as well. Do you have any closing remarks?
Just that multidisciplinary science is wonderful. And, you know, I'm a biologist in a geology department. I took, I guess, I can't remember the number on my transcripts, but I've taken maybe 8 or 10 geology courses and during my graduate years. And I just love the interface between geology and biology. And if your students have opportunities to, you know, collaborate across discipline, it's a really exciting way to do science. And the IODP program is a great avenue for them to get involved. There's so many opportunities, and they can go on that website that's on my talk and get more information. And I know that you guys are not, who are listening, are not asking questions right now in the chat or verbally. But please don't hesitate to reach out to me, you know, if you have any questions, you know, if I can answer them about anything I talked about or about IODP. And thank you so much for listening.
And thank you for participating. I know that we would much rather do this in the Hale Center with a large number of warm bodies sitting there. But unfortunately, this is what we have, and I think it's important. I'd like to thank the Ocean Discovery Program, that you're still one of their distinguished lecturers. Also, the IRIS SSA program that brings us speakers, DB Western and the Mill, as well as the College and College Foundation, Dallas at IT, and also to all of you that have been listening today. We've got at least 5 more talks in a variety of subjects coming up over the course of the year. I mentioned the Cascadia anniversary talk end of January on January 26th, whale seismology. It may not be earthquake and tsunami, but it'll be interesting. Climate change, radon, hydrothermal vents, Kilauea, and who knows, maybe we'll add another talk or two at the end as well. So thank you for your participation. Have a great rest of your day. Thank you.